In 1884, French microbiologist Charles Chamberland invented the Chamberland filter, that contains pores smaller than bacteria. In the early 1890s, Russian biologist Dmitri Ivanovsky used this method to study what became known as the tobacco mosaic virus. At the same time, several other scientists showed that these agents were different from bacteria and about 100 times smaller. In 1899, Dutch microbiologist Martinus Beijerinck observed that the agent only multiplied when in dividing cells. In the early 20th century, English bacteriologist Frederick Twort discovered viruses that infect bacteria, and French-Canadian microbiologist Félix described viruses that, when added to bacteria growing on agar, would lead to the formation of whole areas of dead bacteria. The invention of the electron microscope in 1931 brought the first images of viruses. In 1935, American biochemist and virologist Wendell Meredith Stanley examined the tobacco mosaic virus, and found it to be mainly made from protein. A short time later, this virus was shown to be made from protein and RNA. A problem for early scientists was that they did not know how to grow viruses. The breakthrough came in 1931, when American pathologists grew influenza, and several other viruses, in fertilized chickens' eggs. Some viruses could not be grown in chickens' eggs. This problem was solved in 1949, when John Franklin Enders, Thomas Huckelweller, and Frederick Chapman Robbins grew poliovirus in cultures of living animal cells. Viruses coexist with life wherever it occurs. They have probably existed since living cells first evolved. New groups of viruses might have repeatedly emerged at all stages of the evolution of life. Their origin remains unclear, because they do not fossilize. So molecular techniques have been the best way to hypothesize about how they arose. There are three major theories about the origins of viruses. First, the regressive theory. Viruses may have once been small cells that parasitized larger cells. Eventually, the genes they no longer needed for a parasitic way of life were lost. The bacteria Rickettsia and chlamydia are living cells that, like viruses, can reproduce only inside host cells. This lends credence to this theory, as their dependence on being parasites may have led to the loss of the genes that once allowed them to live on their own. Second, the escape, or cellular origin theory. Some viruses may have evolved from bits of DNA or RNA that escaped from the genes of a larger organism. The escaped DNA could have come from plasmids, pieces of DNA that can move between cells, while others may have evolved from bacteria. Third, the coevolution theory. Viruses may have evolved from complex molecules of protein and DNA at the same time as cells first appeared on Earth, and would have depended on cellular life for many millions of years.